डीएनए को मेम्ब्रेन के ऊपर ट्रांसफ़र करने के बाद जो हाइब्रिडाइजेशन के प्रोसेस तक नेक्स्ट method or next process steps are going to depend upon the type of the membrane, the method of the transfer and the fixation method. मेम्ब्रेन ट्रांसफ़र करने के बाद हमें डीएनए को फिक्स करना पड़ता है मेम्ब्रेन के ऊपर. एंड as we discussed earlier, there are two types of membranes, charged membranes and uncharged membranes. And the method of transfer is going to decide or tell us that what should be next we have to do for DNA fixation. अगर हमने alkaline transfer किया है और हमारे पास membrane है, that is a charged membrane, in that case the DNA which is transferred to the membrane is covalently linked with the membrane. So in that case we have to do nothing. So alkaline transfer for a charged membrane, no further steps are required for DNA fixation. On the other hand, if we have uncharged membranes, then before proceeding further for hybridization, we have to do some steps, we have to perform fixation of DNA. इस पर्पस के लिए हम माइक्रोवेव ओवन यूज़ कर सकते हैं, इसके लिए हम यूवी क्रॉस लिंकर यूज़ कर सकते हैं, या फिर वैक्यूम ओवन में जो मेम्ब्रेन है उसको बेक किया जा सकता है. ये टेबल अगर देखा जाए, दैट टेबल शोज द मेम्ब्रेन टाइप, transfer type, fixation method, and what sequence of steps are involved. For positively charged membrane and alkaline transfer, no further steps are required. For uncharged or positively charged nylon membrane and neutral transfer, we can do UV radiation, we can do baking in vacuum oven or in microwave oven. Depending upon the type of transfer, if this is a neutral transfer, we will have a different buffer. If this is an alkaline transfer, we will have a neutralization buffer too, in which we can soak the membrane. For immobilizing the DNA in the membrane that needs fixation either by baking in vacuum, by heating in microwave oven, or by cross-linking in UV irradiation or by UV irradiation. For fixation by baking in the vacuum oven, we have to dry the membrane. It can be done by wrapping or by covering the membrane with 2 filter papers and then leave that membrane for 30 minutes to get dried on a paper towel at room temperature. Once the membrane is dry, after that sandwich it between 2 filter papers, means on both the sides there will come the filter paper, in between there will be the membrane, and it will be kept in a vacuum oven at 80 degrees centigrade for 30 minutes up to 2 hours. That time is sufficient enough to fix the DNA with the membrane. अगर हमें माइक्रोवेव ओवन का यूज़ करना है, then this needs the placement of damp membrane on a dry piece of blotting paper and then heating for 2 to 3 minutes in a microwave oven at full power, that is 750 to 900 watts. Once this fixing is done, then this membrane can be used further for pre-hybridization and hybridization with the probe. In most of the cases, the UV cross-linkers are available, and in that case, the damp membrane is placed in the cross-linker, UV cross-linker, over a paper towel, and the UV exposure is done for a couple of minutes so that the DNA is fixed or cross-linked on the nylon membrane. Once the DNA is fixed with the membrane, now this membrane is ready to be used for the hybridization with the radio-labeled probe. And in case if we are not willing to use this membrane immediately for hybridization with a probe, it can be stored in the lab, wrapped in aluminium foil or in the filter papers. The membrane should be dry and it should be stored under vacuum. The preference is under vacuum so that the membrane should not get extra moisture, or it should not lose the moisture and get suffered from any kind of problems.